Okay, fine. So we'll begin today's session, and we'll start with the second talk by Professor Arijit Samal. Yes, please go ahead.
So what I plan to do today is basically present basically two stories from my group, which use Boolean modeling of gene regulatory networks. So some of you asked me several questions yesterday about what I do, why I do. So briefly, my research in the Institute of Mathematical Sciences, or my group, broadly works on 4 areas currently. The major area we spend time on is what I call as chemical spaces for drug discovery systems, toxicology, and human exposome. And there we are interested in particularly two types of chemical spaces. One, natural product spaces, in particular phytochemical and secondary metabolites of fungi, which are important from the perspective of finding new drug lead molecules. And a second area we work on is characterizing environmental chemicals which could be harmful to human and ecosystem upon exposure. So that's basically, broadly 60% of my research time. Of course, since my PhD, as I mentioned, I have been working on different aspects of characterizing biological networks. And we do have a sort of a subgroup within my group, which is basically focused on developing methods for analysis of complex, in particular, biological networks. And in that area, we have broadly two subgroups. One which tries to port concepts from Riemannian geometry for use in analysis of networks. And second is what I'm trying to present here, which is Boolean modeling of gene regulatory networks. A third area which we have worked a lot, but slowly we are no longer working because frankly there are not many students who are interested to continue research in this area in MSc because most of the students come from more quantitative background and less biology background. A systems modeling of metabolism and protein secretion system in microbes. These are areas where we lead research projects, and this is the fourth area where we basically play an enabling role in projects which are led by experimental groups either in India or abroad. They typically have some high-dimensional or complex experimental data sets which they want us to look at in a systematic fashion and come up with hypotheses for doing mechanistic studies. So these are some examples of publications which have come out from my group in the last 7 to 8 years in these areas. So I broadly spend most of my time thinking on the first area, maybe 60% and 20% of the time what I'm presenting today, and maybe 20% here. That was also a disclaimer that I may not remember all the time what I'm presenting. So if you ask for details, maybe there are students who may be better suited to answer the questions. So just to recap, I presented a brief overview of Boolean modeling, in particular the different aspects of Boolean modeling from a basic perspective if you are trying to enter the field in the last talk yesterday. So there are a few points I want to recap. So I emphasize that amongst the different frameworks which are available to mathematically model the dynamics of gene regulatory networks, the simplest and perhaps the most elegant in that sense, which is available in particular in the light of paucity of data, is Boolean network modeling. And Kaufman essentially pioneered this field. He proposed random Boolean networks as a means to infer and study the dynamics of gene regulatory networks in 1969. What is important to remember is that Kaufman was proposing the framework in the pre-genome era. So there was a paucity of data. We did not know actually the structure of gene regulatory networks, so approximated it with random Boolean networks where the structure was random. He took some n genes and connected them in a random fashion with a fixed in degree. And also the choice of Boolean functions, as I emphasized, which captures the combinatorial regulation of different genes, was also chosen at random amongst the various possibilities. And what was important is that Kaufman associated the different attractors of the Boolean gene regulatory network to different cell types of a multicellular organism. So that's something he was acutely aware of, and that was the motivation behind his proposition. So this is a one slide recap of what I tried to cover with the basics of Boolean modeling. So I presented the basics of Boolean modeling through this 5-node example. There are 5 genes here, or nodes. Each gene can be in 2 states, 0 or 1. Then there is a directed network which captures the regulation of gene by other genes in the network. And thirdly, the third building block of the Boolean network model is to associate a Boolean function with each gene or node in the network which captures the combinatorial regulation. Once these 3 things are specified, we of course assume a discrete time, and you have to choose a update scheme. For today, whatever results I'm going to present, it will be with synchronous update. And if you take the synchronous update scheme, for this 5-node network, the state space is 2 to the power n equal to 2 to the power 5 equal to 32 states, and you can write down the state transitions for each of the 32 states, and this gives rise to a state transition graph and the basins of attraction of the different attractors. As I would like to remind you, for the synchronous updates case, the dynamics is deterministic and the basins of attractions are disconnected from each other. Okay. What will be important to remember or what I would like to emphasize in the second part of the talk is that note that if you want to model cell-cell transitions, the different cell states would be interpreted as fixed points of the system. In this synchronous update scheme or the deterministic dynamics, there is no way to transition from one basin to another basin, okay, if you want to model the cell state transitions. And later on in the talk, I will present a framework that we developed to allow that to happen by introducing noise into the model. So if there are any questions here, I'm happy to take. If there are any doubts from yesterday on this. So yesterday, after presenting the basics of Boolean modeling, I dwelled a lot about the type of dynamics and the differences in the dynamics that is obtained between synchronous and asynchronous update. And subsequently, I touched upon the problem of choosing or specifying a Boolean function amongst the various possibilities for a given node. So in that context, I emphasized that as the number of inputs to a gene increases, the number of possible Boolean functions, the space of Boolean functions rapidly increases. It is faster than an exponential. And by the time you reach k equal to 5, that is if a gene is controlled by 5 other genes, the number of possible functions is more than 1 billion. And if you are trying to reconstruct a Boolean model from data that best fits the observations, then you have to choose one of the billion possibilities. And you can imagine that even if you know some experimental information, there will be a huge space of functions amongst the billion, a subspace, which can fit the data. So, I also mentioned that Kaufman had no information, so he considered one of the, say, possible functions at random at each node to simulate his model. But he was looking at an ensemble of models and taking the averages inferring the averages as the design principles. But when we are now in the post-genome era, actually creating real models, then we have to actually specify the function which may be at play in the promoter of a gene. So the first part of the talk will actually touch upon that question. And then specifically, we will be asking the following question. I mentioned that in the post-genome era, more than 200 Boolean models have been constructed to date for different biological processes for organisms across different kingdoms of life. Now, if we were to look at those real networks which capture certain biological processes where, modelers have assigned Boolean functions or update rules that capture the combinatorial regulation of a gene, are the rules really random? I wanted to also emphasize that, as we know, those who have almost everybody has taken a systems biology or a network science course, we have realized over the last 20-25 years, after the availability of data associated with the structure of real-world networks, in particular biology, they are far from a Ardha-Shreni graph or a random graph, okay? They are not random. So the question, a parallel question which arises is that if you look at these real Boolean networks, which have been constructed for different biological processes, are the rules random? That is, do you expect any of the possible rules to appear in nature? Of course, we are working with the assumption or coarse-grained assumption of approximating the combinatorial regulation through a Boolean function. So that's basically the first question I will be addressing. And if the choice of functions is not random, then those functions which are overrepresented in the real data, can they be explained by certain striking properties associated with them? So this is a question I'll address. In the second part of the talk, I would be giving a proposal of a systematic framework to leverage the known design principles, in particular the knowledge of relative stability of cell states within the framework of Boolean modeling. That is, how can we capture the metaphor of Waddington landscape within the framework of Boolean modeling for developmental gene regulatory networks. Okay. The work I'm going to present today, these two stories, basically the student who led the work was Ajay Subbaroyan. Ajay was leading both the stories. And in the second story, Priyatosh did significant amount of work, and both the works are in collaboration with my long-term collaborator, Olivier Martin in Orsay. So in the first part of the talk, I will be specifically asking the following questions. And these questions are important in the following sense. Although till today, on to date, most of the Boolean models which have been constructed are of mid-size, say 20 to 30 nodes at best. But the idea is to use the approach for modeling large-scale networks where you may have hundreds of components. So if you look at, for example, the ENCODE data, which gives you the chipset data for various transcription factors in humans, it is very easy to find that there are at least 4 or 5 and even more transcription factors which can bind to a given promoter of a gene. That is, the end degree of a gene in, say, a multicellular organism is going to be very easily 4 or more. While Kaufman, while he studied the random Boolean networks, maybe because of even the computational limitation he had, he studied typically networks with k equal to 2, rarely with k equal to 3. So once you have an end degree of greater than 4, okay, at each node you have at least a billion possibilities, okay, to choose from the functions. So these prompts essentially the following 3 questions. Are certain classes of logic rules or Boolean functions more biologically meaningful than others? In this context, I won't be repeating today. I presented 4 different classes of biologically meaningful Boolean functions yesterday, which we will be taking systematically, with the real data set today. Are certain types of Boolean functions more prevalent in real gene regulatory networks? That is, if we take the data set of functions which can be compiled from across the different models which have been constructed today, are there certain functions which are overrepresented? And finally, if certain types of Boolean functions are more prevalent in real networks, then do they have some striking properties explaining their prevalence? So, to recap, I had introduced these 4 different types of biologically meaningful functions, which were proposed both in computer science literature and also in the literature associated with Boolean modeling of gene networks. So we looked at the effective functions, where every input has to be effective. That is, every input to a gene should elicit some change. Then we looked at the unate functions, which allow you to capture the signs which you may know with each type of regulation. That for each input may be an activator or an inhibitor of a gene. If you want to capture that within the notion of a Boolean function, you can enforce that through the property of unateness. And finally, I also discussed the canalizing property of a function. An input can be canalizing if for one value of that input, that is either 0 or 1, regardless of the values of the other inputs, the output is fixed. So the canalizing property of a node essentially allows you to capture the veto property of a given regulator. And this notion of canalizing function, which was first proposed by Kaufman in his book in 1993, has subsequently been extended to the notion of nested canalizing function, wherein all the inputs satisfy the canalizing property in a hierarchical fashion. So before I go on with analyzing the data, I did not actually introduce another class of biological meaningful function, possibly biologically meaningful function yesterday, so which I would like to present today, and then we can go with the results. So this is a class of functions which is known in the computer science literature as read-once functions. I must mention that the four types of functions which I had covered yesterday, before our work, others had also studied in the context of Boolean modeling of gene regulatory networks, but the read-once functions before our work had not been explored in the biology literature at all. So we actually ported this from the computer science literature for the work in gene regulatory networks. So a read-once function is a Boolean function wherein if you write the expression of the Boolean function, each of the input variables only appears once in the expression. And the variable can appear either as a positive literal or a negative literal, that is with a NOT gate. And you basically combine these inputs through either a AND or OR gate. It's a simple expression. So as it will turn out, the read-once function captures all the NCFs. That is, NCF is a subset of the nested canalizing functions happen to be a subset of read-once functions. So in the read-once functions, actually when you write these variables one at a time, there is no restriction on where you put the braces. But if you look at the canalizing function, the braces appear in a hierarchical fashion. So the read-once function actually contain two subtypes of function. One which is the nested canalizing function, and the second which can be called as non-NCF ROFs. So there are certain properties we can prove for them theoretically. One is that the read-once functions are such that they always have an odd bias. That is, if you take any function, any read-once function, write its truth table, on the output vector there will be only odd number of ones, never an even number of ones. They happen to be all effective. That is, it's a subset of effective functions. They happen to be unit, and more importantly, the class of read-once functions and subsequently nested canalizing functions, they occupy a very small space within the space of all functions. For example, as I mentioned, there are 2 to the power 2 to the power 4, 6, which is equal to 65,536 possible 4 input functions. Of these 4 input functions, the number which is read-once is 832. And if you ask, the number which is NCFs is roughly 600. So as you can see, it's a very small fraction of the complete space of functions. So we have these 5 different classes of functions, okay? And let's look at how they are organized in the space of possible functions. And we can view this by taking the, all possible functions at 4 inputs. Why 4 inputs? Because there are 65,536 functions. It's easy to, evaluate each of the 65,536 and figure out which class they fall into. So as I mentioned yesterday, all these 65,536 functions can be divided into first 2 sets. One which have the odd bias, and the second which have the even bias. All the ineffective functions are lying on the even side. Then you have the canalizing functions which can be both even and odd and even. The unite functions which can be also odd and even. The read-once functions only occur in the odd bias, okay? And the NCFs, which is a subset of read-once functions, also are in the odd bias. Any questions till now? I think this should be absolutely clear. So the most important thing I wanted to say is the following. Let's try to ask the following question. As we increase the value of k, okay, what fraction of the possible functions which are there occupy the space of, say, nested canalizing functions and read-once functions? We have done it for all the different classes we have studied. So as you can see, at k equal to 5, the fraction of possible functions which is read-once is 10 to the power minus 6, 3 into 10 to the power minus 6 roughly. So there were billion functions. So if you multiply by 10 to the power minus 6, it is roughly in some thousands. Okay. So from the space of billion functions, you have come down to only 1,000 functions. So the most restrictive class happens to be NCF, and you can actually do this plot for K much higher, and you can see that. So if you go from K equal to 1 to 2 to 3 to 4 to 5, the absolute number of nested canalizing functions is increasing, okay, but the fraction of the possible functions is extremely small of the all possibilities. That's clear? So as I mentioned yesterday, in the post-genome era, subsequent to genome sequencing, which started towards the end of the last century, people have started constructing, because of the available information on the parts list of an organism and also the interactome, Boolean models for different biological processes. So essentially, based on the experimental literature, you get the biomolecular components, you can get the regulators of nodes, you can infer the logical update rules from ChIP-seq data, RNA-seq data and other experiments, and you can get the steady state attractors based on the expression patterns, which can be quantified with the different approaches like microarray to RNA-seq to single-cell RNA-seq, and this have led to the reconstruction of various Boolean models. As things stand today, there are more than 200 models which have been published for different processes from development to cancer for different organisms. And when we did this work, and we were doing this work in late 2020, my student Ajay essentially compiled 88 of those models at that time. And these 88 models, if you combine them, cumulatively is a corpus of about 2,687 Boolean functions. These 88 models can be classified into differentiation and development. 28 were such, 20 were cancer models, 11 were signaling pathways, immune response, cell cycle, and metabolism. Doesn't matter. This is a large corpus of functions. Okay, it's of course not exhaustive, but it is a large enough sample to ask the following questions: are Boolean functions in real networks random or not? Okay.
So just curious, what's the average number of nodes in these empirical Boolean networks?
We have a distribution, but these are all small. Typically, some models are 4 and some go up to 30, 35.
All right. So these nodes are identified from experiments that they are involved in this particular function.
Yes.
I see.
Yes. But in my PhD, I studied a network which is not in this corpus, which has about 500 nodes. So I don't. One of the reasons they restrict to, so even if you see Mohit's model is 32 nodes only. There's a reason for that. If you want to exhaustively sample all initial conditions to find the attractors, you are limited by 30 because your state space is 2 to the power 32. You will typically not find models with more than 30. If it is more than 30, then there are some approximate algorithms to find what are steady states. But people typically try not to do that. And these models have also the other property that they take the core component of the model, wherein every node has an in degree and out degree. That is, the things which have no in degree and things which have no out degree are removed actually. That's why they're typically small. But the model which I studied with 500 nodes, the core was very small. There was a lot with no in degree and no out degree. They don't actually are that relevant for the dynamics, if you can. So essentially, we compiled this corpus of 2,687 functions, and this is, to date, the largest such study to be conducted. It was a lot of effort because Ajay also had to read a lot of papers and, compile each interaction and the model and all that, science of the interactions, and it had to be manually done. So, as Sitabro was asking, a related thing to ask is that we had this 2,687 functions. You can actually sort of divide these functions into based on the number of inputs there are to each function. Okay. So this is sort of the histogram of this sort of dividing the functions into different number of inputs. So there was a huge number of functions with only single input. There were many functions with two inputs, and as you go down, you rarely find functions with many inputs, but actually in the data set there was even one function with 14 inputs. Okay. So the first question we asked is the following, which is a very simple question. I don't think anybody had asked this. So I showed in that sort of a two-dimensional graphic with the schematic, that half the functions half the functions are odd bias, half the function are even bias. So I was curious, how probable is to find that? If you choose randomly a function, then you have equal probability of finding an odd bias function and an even bias function. But when we looked at the real data, so the green are the odd functions and the red are the even bias function. Overwhelmingly, almost every function was an odd bias function. Of course, later on it will become evident why this is expected, but this was clearly a surprise. And, funnily, I quote that living beings seem to favor odd biased regulatory logic. In the next step, what we asked is that after, of course, you can do a statistical test, which we have done in our paper, and I'm not going to present those, intricate details. Odd bias functions are overwhelmingly significantly enriched in the data set. So next we asked, we had all these different classes of biologically meaningful functions: the effective, unit, canalizing, nested canalizing, read-once functions. Are they enriched in the data set? So of course we do it for different number of inputs. So the histogram bars are essentially the values in the real data, that is the corpus of 2687 function, and we present it as the fraction of the possible functions. And the black dots here are what you expect if you chose at random. Okay. So if you ignore the effective functions, so here's something I wanted to let you know. As k increases, almost every function is an effective function. Hardly any function is ineffective. Okay. So choosing amongst the effective function is almost like choosing a random function. If you ignore the effective case, all the other classes are, unit, canalizing, read-once, and nested canalizing are enriched in the real data. Okay. You can see the difference between the black dot and the bar. Okay. We have done a statistical test to show that it is really enriched. So a related question we asked, for which I didn't prepare a slide because it's a much more intricate detail, is that I told you that, of course, the NCF is a subset of canalizing function, and an NCF is also a subset of read-once function. Fine. So now, when you see this enrichment for these four classes, you may wonder if the enrichment is only due to the superclass or superset rather than the subset. That is, are we seeing this enrichment because nature is going for unit functions? Because unitness is a property which naturally has to be satisfied, activation and inhibition. Or, NCFs are unit, ROFs are unit. So we see the enrichment for ROFs and NCFs because they happen to be unit. Or is there some property associated with NCFs and ROFs beyond being unit that they are enriched? So we have developed a relative enrichment test in this paper where you can ask if beyond being a unit, ROFs are still significantly more enriched. I don't want to go into the details. The result of that analysis is that there are properties associated with read-once functions and nested canalizing functions which are beyond being unit that they are enriched. Okay. And furthermore, I can say that there is an enrichment for nested canalizing function even beyond ROFs, why they are enriched. Okay. So these subclasses seem to have some property which show specific enrichment. So the next point we wanted to address is that in particular we found that the NCFs and the ROFs were enriched in the data. So we asked the question, could there be some striking properties associated with these class of Boolean functions which could explain their enrichment? And for that we draw motivation from what Kaufman said in his seminal book in 1993. So Kaufman in his 93 book, The Origins of Order, conjectured that the Boolean functions that capture the regulatory mechanism may possess, in his words, quote-unquote, chemical simplicity. In particular, he had conjectured that the canalizing functions, not the nested canalizing functions, might be a class of Boolean functions that enable this simplicity. So we interpreted this simplicity in terms of minimum complexity. And complexity is a term which is used often, but you need to quantify the complexity in some way. So we decided to quantify the complexity of functions using notions which we borrowed from computer science. And we essentially used two metrics. You could use other metrics as well to quantify the complexity of functions. And the first metric we used was what we call as Boolean complexity. And this concept was ported from a seminal paper in psychology, actually in Nature in 2000, where Feldman actually used this to try to check the complexity of different decisions that a human mind takes. So it was actually not even from computer science, but from a psychology literature that we ported this concept. So what is Boolean complexity? Feldman defined Boolean complexity as follows. It is the number of literals in a minimal equivalent Boolean expression obtained by factorization of the Boolean expression. So let me try to give you a feel of how you compute the Boolean complexity. So this is the disjunctive normal form for the Boolean function, one Boolean function. So we have 8 possible rows in the truth table, and the output value of each row can be 0 or 1. Okay. So the DNF basically has one expression it's a combination of ORs across the rows which give you 1. Say so let's say these are the 3 rows of the truth table which give you 1. So the DNF will be as follows. It is x1, x2 bar, x3 plus, so this is a Boolean OR, x1, x2, x3 bar plus the last is all 1. And that's basically gives you the F. Clear? Now, to get to Boolean complexity, you have to simplify the expression to get the minimal equivalent expression. And what I've shown you here in the 2 steps there are how you simplify that using Boolean algebra. So in this case, what you can do is that you can first take out x1 bar x2 between these 2 terms, and you have x3 plus x3 bar. All of you remember the basics of Boolean algebra. It is 1 or 0, which is always 1. So that gets, this is 1. And then you get x1 bar x2 plus x1, x2 bar x3. This can be further factorized to a simpler form. And the number of literals that appear in this expression are 5. So the Boolean complexity of this expression is 5. So what is important is that computing the complexity by getting the minimal equivalent expression is not straightforward. As k increases, getting the minimal expression is not guaranteed. There are some heuristics available to approximate that, but it's a challenging problem. So I won't go into the details how we do that, but we can actually get a sort of a lower bound or upper bound for each function. So this is one of the metrics that we use to quantify the complexity. Okay. So if your Boolean complexity is less, then you have a much more simplified expression to capture the function. The second measure of complexity that we use in our work is known as average sensitivity. And sensitivity itself as a measure has attracted a lot of attention with respect to Boolean functions in the computer science literature. And average sensitivity had been used in the perspective of robustness to perturbations in the literature on Boolean modeling of gene regulatory networks already. So basically, the average sensitivity captures the probability of observing a change in the output bit if a single input bit in the Boolean function is flipped. So how do we compute the average sensitivity? So I had introduced the Boolean hypercube representation of a Boolean functions yesterday. And one of the reasons is because I wanted to show sensitivity and subsequently a proof down the line using this representation. So you actually compute the average sensitivity as follows. You first compute the sensitivity of each vertex in the Boolean hypercube, which is basically for each row in the truth table. And it is done as follows. So in this Boolean hypercube, you just have to ask for each vertex how many of its neighbors have a different color. So the color corresponds to the output value. So for example, if you see this 0, 0, 0, it's blue. It has three neighbors, 1, 0, 0, 0, 1, 0 and 1, 0, 0, 1. Of the three neighbors, only one of them have a different color. So the sensitivity of this vertex is 1. And we do this for all the 8 vertices of the Boolean function with 3 inputs. So I am just telling you the number. Okay. So you get 8 numbers and you can add them, which gives you the total sensitivity. So the total sensitivity is 14 by adding those numbers. And then you average it by dividing by the total number of vertices in the Boolean hypercube. Okay. That gives you the average sensitivity. So remember that what is it capturing? So essentially, you're trying to see for each vertex, if you flip one of the nodes, one of the states, okay, then do you go to a different output value? So if you don't, then your sensitivity is low. That also means you are robust, because if there is a noise which flips the gene state, then you are unlikely to go away to a different output. So the average sensitivity for this function is 1.75. Is that clear? And computing average sensitivity is straightforward for a Boolean functions, unlike this measure of complexity, which is Boolean complexity. Any questions till now? Oh, very good. So what we then tried to do is we took all the 4 input functions, the 65,536 functions, and we wanted to study if there is any relationship between Boolean complexity and average sensitivity, just empirically. And actually we made a 3-dimensional plot where we partitioned the functions into also the bias. Okay, as I mentioned, you can have bias values between 0 and 2 to the power n minus 1. 2 to the power n. So I won't go into some of the details, like there are some complementarity which we exploit and so on. But if you look at this, so if you look at the shapes of these points, they signify different types of functions in the data. So this is a 3-dimensional plot for all the 65,536 functions, where we have plotted the average sensitivity, Boolean complexity, and the bias of a function. So I take a 2-dimensional projection of this 3-dimensional plot and first try to see if there is a relationship between average sensitivity and Boolean complexity of a function. You can see there is a very nice linear relationship, and the correlation between the two is as high as 0.812. This is only an empirical result. Will this be true for other values of k? That is, do we get high enough correlation for k5, k6? We don't know. It's very difficult to do because there are at least a billion functions at k equal to 5. At k equal to 4, we could do it, okay, because there are only 65,536 functions were there. So there is clearly a correlation between average sensitivity and Boolean complexity for 4 input BFs. Okay. So then what we decided to do is to plot from the three-dimensional plot Boolean complexity with bias. And remember, the functions which we found to be enriched were the odd bias functions, okay? So the bias values which are even for all perspective don't matter here. And I also told you that you are always, NCFs and ROFs are odd bias functions. So when you look at the odd bias values, okay, here, you can see that the functions which have the least Boolean complexity for a given bias, okay, is always a NCF or a ROF, okay. I mean, it's very simple because look at the definition of a ROF. It's a function where each variable appears only once, okay, in the minimal expression, right? If you are effective, each variable has to appear once. So by definition, ROF is the class of functions which minimizes Boolean complexity, and NCF is a subset of it, okay? So essentially, I mean, we see the enrichment for ROFs, and it's very simple explanation for the enrichment of ROFs and NCFs is that it minimizes this notion of complexity, which is Boolean complexity. Is that clear? Yeah, Choling.
So I can get an intuition about sensitivity. So can you sort of give us an intuition as to what complexity would be? And then in your previous slide, you showed that there's sort of a relation.
You mean this one, this notion?
Yeah, Boolean complexity in general. What am I supposed to see when?
Okay, so any function you have to write an expression, right?
Sure.
The simplest expression wherein each input has to appear at least. So the input will be effective, it appears in the expression. So actually, in terms of the Boolean expression, when I say input is ineffective, then actually that variable doesn't appear in the minimal expression. Okay. So each variable has to appear in your expression because it contributes, right? Now, ROFs is the simplest expression wherein each variable appears once.
Right. So in dynamical systems, we sort of differentiate between what's complicated and what is complex. So I don't suppose, complexity here is defined in that spirit for dynamical systems.
Here, the complexity is, I'm not trying to quantify the complexity from a perspective of dynamical system, but in terms of the logical update. So in terms of the combinatorial logic, I want to minimize the complexity. Because Kaufman thought,
or let me ask you this: so what would be a complicated logic then? Do you have a sense of a complicated function? And would you differentiate between a complicated function and a complex function?
the point is that it seems that at least the data that we have analyzed, everything seems very simple. So we don't encounter such complex things, okay? But I can construct one if you want, but I have not so. So there are many fascinating things with respect to this bias, and nobody has actually explored that so far. Again, this bias perspective, which I'm trying to avoid many things because many details which are in the paper, this bias perspective before our work had never been taken.
I know, but I was just fascinated with that relation that you showed between complexity and sensitivity.
That's not no.
So I'm sorry.
That's just an empirical observation. So I don't even know if that will stand the test of time if we go to k equal to 5, 6, a 7. Maybe it will just decay off the correlation. We don't know that actually. It's an open problem.
Yeah. Well, thanks.
So, okay, just to recap, the ROFs and by definition its subset NCFs minimize the first notion of computational complexity, which is the Boolean complexity, right? But when we looked at the real data, I told you that we found an enrichment of NCFs, which is beyond ROFs. That is, NCFs have certain property, it's likely to have certain property beyond ROFs, that they are even enriched further. So then we wanted to see, could there be some other perspective which distinguishes between a ROF and a NCF, okay? In particular, the subset which is the NCF. So, I mean, this is something we were doing in an empirical sense. So then we plotted the average sensitivity as a function of bias. And I should have circled here. So the squares here are the ROFs, that is non-NCF ROFs, which are not NCFs. And the triangles are the NCFs, okay? So look at the bias 7 here. If you see the average sensitivity, so if you look at all the odd biases, you will see the minimum value of average sensitivity for any function is a triangle. So this is an empirical observation that for any given bias, the least value for average sensitivity is seen for a NCF. In particular, at bias 7, we find that the NCF has a lower average sensitivity than the non-NCF ROF. That is, you have two types of ROFs there, NCF and the non-NCF ROF, and the non-NCF ROF has a higher average sensitivity. And this we were able to computationally check for k equal to 4, 5, and this led us to conjecture the following, and this is just an empirical observation. So we don't know if this is true if we go from k equal to 4 to 5, okay? For that to be checked, it's difficult because you have a billion functions. So we asked the following question. So this led to the conjecture that if you fix k and bias p, then NCFs will have globally minimum average sensitivity. That is, the fraction of functions within the ROFs which are NCFs, they will always have lower or the lowest average sensitivity possible for a given bias. Now, this is a conjecture, and this is a conjecture made for k, any value of k, not just 4, which we have computationally checked. So we basically then provide an analytical proof that this is true in the paper. And I'll try to give you a feel of the analytical proof rather than going through the proof in detail. So essentially what we are saying is that NCFs minimize the average sensitivity for a given bias, okay? So what does that mean? So let's look at the Boolean hypercube representation of a function, okay? So what we are saying is that you have a given k, the number of inputs, that decides the size of the cube. In this case, it's a 3 cube because it's a 3 input function. And you have a given bias p. Bias p means the number of ones in the truth table, okay? That basically means how many of the vertices will be of red color, okay? And we want to minimize the average sensitivity. So the average sensitivity is essentially a normalization over the total sensitivity. So we want to minimize the total sensitivity. It is easy to show that the total sensitivity of a function is basically 2 times this quantity E01, where E01 basically signifies the edges, the number of edges in the Boolean hypercube that connect, two different colors. Okay. So if you see here, these are the edges which are now shown in red, that connect blues and the reds, okay? You count basically the edges that connect 2 different colors on the Boolean hypercube. Take the twice of that is basically the total sensitivity. So minimizing average sensitivity is equivalent to minimizing this total sensitivity, which is equivalent to minimizing the E01 value. So which basically can now be recast as a problem in computer science. So if you are given a bias P, how do you arrange your P vertices on the hypercube such that this number E01 is minimized, okay? So we could cast the problem to a equivalent computer science problem where we wanted to find an arrangement where this E01 is minimized. And it turns out that this problem, of course, I guessed would have been solved in computer science, and it has been solved in various contexts. One of the best expositions of this is by Sergio Hart in this paper, where he has proposed a certain arrangement for a given P, which will minimize this E01. Okay. So I won't go into the details. So that was solved in the computer science. The only thing then we had to do is that the arrangement that the computer scientists had proposed, which would minimize the number of edges between the 2 colors, we were able to show that all such arrangements with odd P, they were equivalent to a nested canalizing function. Okay. That's the only thing we had to do, in which we showed visually. This is shown in the paper. You can happily look at it. We have a visual proof of it. A detailed analytical proof is also available in the supplementary material, not important. So for example, if you had to arrange 5 vertices on a 4 cube such that you minimize this E01, this is the arrangement that is given. Okay. I won't go into the details. Okay. So we could just cast the problem as a problem on the Boolean hypercube, then to a problem of finding the minimum of edges between 2 colors, and then we got the arrangement from solutions in computer science literature, and we just had to show that arrangement is always a NCF. Okay, and with that we have proven that for any value of k and bias p odd, the NCF is always going to have the minimum of average sensitivity. So to summarize, so this is the analytical proof which holds for anything. To summarize the first part of the talk, are certain classes of logic rules more biologically meaningful than others? We have presented some: effective, unit, canalizing, read ones, and NCFs. Are certain types of Boolean functions more prevalent in real gene regulatory networks? It turns out that 2 classes, NCFs and ROFs, are in particular. And if they are prevalent, are there certain properties associated with them which could explain, plausibly explain their preponderance? We show that the set of ROFs, which includes NCFs, minimizes the first notion of complexity, Boolean complexity, and NCFs, which is a subset of ROFs, also minimizes another notion of complexity, which is the average sensitivity. Okay. So that's the story in the paper. So I'll make brief comments in the next 2 slides. So those who come from the dynamical systems perspective, a question which must be bothering you is that now, if NCFs are preponderant in real data, do they have, or do their presence have, a dynamical consequence? Okay. So in the paper, in this paper already, we show or we give a insight towards that. So actually, this average sensitivity is defined for each function, and you can actually compute an average of the average sensitivities of all functions across the nodes of the Boolean model. So some interesting work by Brian Daniels and colleagues have shown that this network average sensitivity can be related to the type of dynamics that you observe in Boolean case. In particular, if your network average sensitivity is close to 1, you are at the edge of chaos, which is at the boundary of ordered and the chaotic regime, which Kaufman hypothesized to be important for living systems to exist. So in short, what we show is that when you actually have NCFs and ROFs in your dynamical system, you are likely to be at this edge of chaos, which the real systems happen to be, over if you choose randomly a function, even in effective unit or canalizing. So in recently a work which has been co-led by my student Priyadosh and Ajay, this is a work I'm not going to present today, where we have taken this further in a much more detailed fashion. Here we ask, do the choice of functions actually lead to certain features in the state transition graph? In particular, we were interested in 2 features, which is the bushiness of these state transition graphs and also the convergence rates. So here is the network which is fixed in architecture, and we also fixed the set of attractors, that is the cell states. You can choose NCFs or you can choose randomly other functions. As I said, all functions are effective in practice. You get the same attractors in the state transition graph, but the state transition graph, when you choose NCFs, looks like this. It has a more bushy structure in the sense that there are many states. It's in synchronous update. Many states in this state transition graph which have no end degree, where there are much fewer such states here. And when you see this contrast in the state transition graph, you can imagine that the dynamics could be very different as well, although you have the same attractors. And what we show is that using 2 quantities, which I don't want to define here, G density and a convergence rate, when you go for these biologically meaningful functions, that is NCFs, you have an increase in both bushiness and convergence rate. So to summarize, biologically meaningful functions result in also higher bushiness and convergence, indicating higher dynamical robustness. So this is something we have shown in this paper. It just came out last month in Briefings in Bioinformatics. It's a very detailed study. So if you want to look at how the choice of functions could influence the dynamics, or the type of dynamics, that we have characterized it already. Okay, so I have about half an hour and I have about 20 slides. So any questions till now? Otherwise, I am going to move to the next story. So the take home are that there are certain types of functions which seem to be enriched in real data. And those functions occupy a very small space of the all possible functions. They seem to have certain striking properties, especially with respect to the complexity notion in computer science. They seem to be amongst the simplest functions which could exist in nature. Okay, if you were to limit yourself to the coarse-grained view of Boolean logic. And further, choice of such functions has dynamical consequences. Okay, in particular towards pushing the dynamics towards ordered regime, and also other properties like high convergence rate and robustness, dynamical robustness, that you desire of a biological system, where you mostly desire homeostasis in most cases. Okay. So let me go on with the second story now, where we propose a systematic framework to leverage the known design principles, in particular the relative stability of cell states or the developmental landscape for Boolean model selection. So of course, in dynamical systems theory, and over the last 100 years or so, 80 to 90 years, people have thought of modeling the gene regulatory network as a dynamical system, and many people have now subsequently viewed the different cells fates as multi-stable states of a dynamical system. To the best of our knowledge, the first person to think on these lines, wherein he posited that cell types could be, seen as multi-stable states of a dynamical system, is Max Delbrück. I think it has been already mentioned in this workshop. And subsequently, in 1957, Waddington conceived his idea of the landscape, where he viewed differentiation as a ball rolling down a sort of a landscape. At each bifurcation point, it commits to one of the cell types and so on. Subsequently, René Thom in 1968 developed a mathematical formalism inspired by the Waddington landscape, to capture this landscape. I mean, this is leading up to the work of Kaufman. I wanted to just give you a sort of a historical, chronological way, if I may say so, to where Kaufman existed. So, and Kaufman, of course, gave the systematic framework in 1969 of Boolean modeling to capture, the dynamics of gene regulatory networks, and which allows us to view multiple attractors of the system as different cell fates. Of course, this was all theoretical work then. And at the start of this, century, towards the end of the last century, in particular, Sui Wang and Donald Ingeber. So if you remember, Mohit showed that, sort of the developmental landscape, where at each bifurcation point he was putting a toggle switch, which was essentially the paper by Sui Wang and Joseph Zhu. So what Sui Wang can be credited with in particular is that he actually connected these with experiments and interpreted also the attractors of the Boolean system, not only with different differentiated cell states, but also cellular phenotypes. And he actually provided experimental support for this idea. And this was very important because subsequent to that, there has been a lot of interest in, using Boolean modeling for example, studying the cell state transitions, Boolean models of cancer, and so on. So essentially, we want to capture the metaphor of the Waddington landscape in our modeling framework. Basically, what Waddington envisioned is basically the development of a zygote is kind of a ball rolling down a landscape. The less differentiated states are at higher altitudes, more differentiated states are at lower altitudes. One needs to remember that Waddington only thought of differentiation, but subsequent to the work of Gordon and Yamanaka, we need to think of also de-differentiation, trans-differentiation, and cellular reprogramming in this picture. That is, there are not only transitions from higher altitudes to lower altitudes, but you also want to view the transitions given the right sort of a perturbation from lower to higher altitudes. Okay, so this landscape picture has got a bit of a modification in this modern scenario. Okay, so when we are trying to capture this metaphor in our Boolean model, we need to capture this modified landscape metaphor. So the basic Boolean model, where we have synchronous update, and even in the asynchronous update, it has the following limitation. So I took this 5-node network with functions, we took the state transitions, and we drew the state transition graph. We had 5 attractors, 4 of them are fixed points, which are relevant for the kind of developmental states we want to model now. So if you see, these bases of attractions are disconnected from each other. And we want to model the cell transitions. So since the basins are disconnected, how do we make them connected? Okay. So in 2016, this paper was published, but actually I was working with Joseph Zhu. I mean, Zhu and Huang are the same, actually the same. Joseph, actually my friend, who wrote the paper with Sui Wang, and this is a work also done in collaboration with Sui Wang. So we started thinking about one of the reasons we were working on it, that it was 2013 or 12-13, and Yamanaka had just got the Nobel Prize. And an important question which was bothering everybody is that how do we increase the efficiency of cellular reprogramming? Okay. So, and it was thought that by modeling these systems, perhaps we can get ideas on different types of perturbations, other types of perturbations, which can improve the efficiency of reprogramming with respect to the induced pluripotent stem cells. And in that context, Joseph and me started discussing how we can exploit Boolean modeling for this purpose. And at that time, we realized that, for example, to model the cell state transitions, and in particular, if we want to predict different trajectories from going to one attractor to another attractor, this deterministic dynamics has a limitation. And we came up with a very simple systematic framework to turn this synchronous deterministic dynamics into a stochastic dynamics which allows such transitions provided the right type of perturbation. So we introduce a noise eta, and basically this had also has been proposed by Kaufman, not this type of dynamics, but essentially he said that any intrinsic or extrinsic noise beyond the delays, the synchronous versus asynchronous update, the kind of perturbation it will induce would be to flip the state of certain nodes. Okay. So we have to somehow flip the states to go from one basin to another. So what we do is that we introduce a small noise eta, which allows with a small probability to take one state, in particular maybe the attractor, to another state in the other basin. Okay, and then you can have such transitions. I must mention that you can interpret this noise eta as both an intrinsic or extrinsic noise. Okay, the details are not important, but the probability that such transition happens is a function of the Hamming distance, or how different are the two states. For example, the state 12 and 20 require two different flips. Their Hamming distance is 2. So the probability is proportional to this Hamming distance. So if the states are very similar, that is only one flip is required, the probability is higher. If it's more than one, as we require more and more flips, the probability decreases. Okay. So I don't want to go into the mathematics of too much of this. So this is actually the transition matrix for the deterministic dynamics. So the matrix T is such that the element Lm corresponds to the transition from a state M to L. Okay, not L to M, but M to L. So of course, each column will have only one entry as 1. Because, in the deterministic dynamics, we know there is only one successor of a state. Okay. Then we have this noise term, okay, where any state can go to any other state. But we give the probability of 0 to the state to go from the same state to another state. And that's defined by this noise term. And typically we do simulations with 5% noise. Okay. And then you can combine them to create a transition matrix T star in a way that this is now stochastic. Okay. That is the probabilities on the columns add up to 1. And this matrix now is ergodic. That is, there is at least some infinitesimal or small probability of transitioning happening from any state to any other state. Okay. Now that we have built this stochastic matrix, now you can start envisioning the cell state transitions using this model. Okay. The other thing we did is that in that paper, this is more a theoretical paper because we did it with a 5-gene network at that time. We proposed also a measure to quantify the relative stability. That is, how do we quantify that the stem cell is less stable than the differentiated cell, okay, in the Boolean model. And for that, we proposed a measure which is there in statistical physics, the mean first passage time, to capture this relative ease of transitioning from one state to the other. So essentially, if 8 is less stable than 13, okay, then it will take less time to flow from 8 to 13, but it will take much more time, even if the right perturbation is given, to go from 13 to 8. Okay. Fine. So we could quantify that. So you can now see that 8 is less stable than 19 and so on. So you can, based on this landscape, you can have pairwise ordering of stabilities. Okay. So why I'm saying this pairwise is because when we are actually trying to build a model using the data from experiments, we may not have complete information. Maybe the system that you are modeling may have 5 states, but you only know the relative stability between the stem cell and the other differentiated states. That is, you have incomplete information. But because we have this, you can model these pairwise relative stabilities, you can still go ahead and incorporate the incomplete information into your model using this framework. So I had stopped working in this area subsequent to 2013 till 2020. After our proposition of the mean first passage time, there have been other measures which have been also proposed to quantify this relative stability of states. And we have actually formalized these measures which have been proposed in a systematic fashion in our recent work. So let me not tell you about all the 5 different measures, but let me just tell you the 2 which may be most important to understand what I'm going to tell next in the talk. So one of the simplest measures to quantify the relative stability could be to see the size of the basin of attraction. So if you look at this, this is the basin of attraction for the fixed points which were observed with the 5-node model. You can clearly see that this has many more states flowing into that, okay? So that has to be more stable, okay? So one of the simplest measures to quantify the stability of the state is just to look at the number of states which flow into that, or the size of the basin for each, okay? And you can actually formalize that with a formula to quantify the stability. So actually this measure actually works pretty well, as I will show later on. But what is the advantage of with MFPT is that you can, in addition, get even prediction of which are the possible trajectories through which you can go from one to the other. Okay, and that is why it is, in terms of the 5 measures, this is perhaps the most suitable for the purpose at hand, and that is why we proceed with that. So we take a case study to show how this could be useful. Okay, this framework could be useful. So we took the model plant Arabidopsis thaliana. The reason we took this, that in particular for a part of the development or a development of a particular plant part, there have been several detailed models which have been proposed over the date. I don't want to go into the details of the biology here, but in particular, the group of Alvarez Bula, she has been working or since 2010 at least, and has proposed more than 4 models with increasing complexity and details over the years on this system. They focus on the development at the root apical meristem. This is, let's forget about the details. So if you see, this is the tip of the root. There are perpetual embryonic stem cells there, which are in the meristem, which keep dividing and allow the plant growth to happen. It's the primary growth of the plant. And, the pool of these cells, which are known as quiescent center, they go through typically asymmetric division into different initials, which then go and form different parts of the root. Okay. So, the model which we started with is the 2010 model, their first model. They captured 4 of these cell types. Okay. Quiescent center, cortex endodermis initials, vascular initials, and columella epithelial initials. Over the years, they have made this model more detailed by increasing the number of nodes, that is the components they capture, with newer interactions and also different cell types. So this is the 9-node network, which they have constructed, okay, which captures these 4 different cell types. Okay, so these are the steady states. So each node can have value 1 or 0. These are the attractors of the system. These are supposed to be the fixed points for this model, which they proposed for the root development. So I may have to use the board here. So one question to ask is that, what we wondered, which is related to the first part of the talk, is that they have assigned a particular Boolean function for each of these nodes, which they have interpreted based on the experimental observations. So the first question to ask is that, are they the only functions which would have given you these attractors and the dynamics? Okay. So if I was to not care about these 4 attractors and ask, let's say I fix this network architecture, that is the set of interactions is fixed and the nodes is fixed. How many different combinations of Boolean functions or models I could construct? So that's a very simple number. So look at this node, JKD. Okay. It's controlled by 2 other nodes. Okay. So since the k is 2, the n degree is 2, the number of possible functions is 2 to the power 2 to the power 2, which is 16. This is only has 1 input. So you have some things which 5 or 4 inputs also. You can multiply this number 16 with the number you obtain for each of the nodes to find the not required, sir, not required, to find the number of possible functions combinatorially across the nodes. And that's actually equal to 10 to the power 21. So if this seems a simple network with I think 9 nodes and maybe about 15 edges, already the combinations of Boolean functions which are possible is 10 to the power 21. But you said, okay, that did not take into account that we get the attractors, okay, the 4 attractors which you desire. I don't want to go through the method we have, which is very small trick we have. We have a method to figure out the subset of Boolean functions or the combinations of Boolean functions which can actually give you back the same attractors. So if I was to impose this constraint that I fix this architecture, I desire that these 4 should be the attractors. There could be other attractors, but these 4 are clearly the attractors of the system. What is the combination of or the set of Boolean functions which give you that different combinations? Actually, the different combinations which is possible is 5.63 into 10 to the power 14 for this model. Okay. So till now, I have not imposed any class of function or biological meaningful function which we learned in the previous story, which we see enriched in the real data. Okay. So the first question we ask is that, can we leverage the design principles, that is the overrepresentation of certain class of functions, to restrict the space of possibilities, okay, of the biologically plausible models? So I'll try to give a schematic illustration of how this space will go. So let's say So let's look at the subspace of all realistic Arabidopsis thaliana root stem cell-ish Boolean models using biologically meaningful regulatory rules. I will keep the structure fixed, okay, and but just limit myself to the different types of functions. Remember, the model also gives you the positive and negative inhibition of each edge. So all functions 10 to the power 21. But let's say I was to limit to only effective functions. Then I can go back to down to 10 to the power 19. If now I say I want the 4 attractors to be given by each of the mod combinations, then I come down to 5.63 into 10 to the power 14, which is close to 10 to the power 15. Then I limit myself to unit functions, that the signs that each node are complied with. Then you can see that this is the space of unit functions 10 to the power 14. It's the intersection of this and this which you will restrict yourself. Then next so the signs are also conforming. Then I restrict to read once functions, NCFs. And if you were to restrict to NCFs, which conform to the sign and to the 4 attractors, we get back actually from 10 to the power 21 to 10 to the power 14 to only 1275 models. Okay. So as you can see, we can get down to a manageable number by sort of leveraging the design principle of nested canalizing functions. So till now, I have imposed only the following constraint, that we have a certain type of function and we also have we get back the same attractors. So this basically leads to a substantial reduction in the number of biologically plausible models. But the next thing we do in this work is, I'm giving you the spirit of the work. The details can be gotten even if I skip some of the slides. Then we wanted to ask that, okay, this is good, but if you have a developmental gene regulatory network, it should capture the metaphor of the Waddington's landscape, right? Your stem cell should be less stable than the differentiated one, right? So do all 1275 models satisfy that? We don't know. Can we put in that constraint to further restrict the space of models? Okay. So it turns out that if you want to first ask the question, which of the 1275 models only give the 4 attractors and no other attractors, we can get down to 170 models. And then you basically put in the constraint, which we know that the quiescent center is basically the least stable cell state. Then you can go down from 170 to 80 models actually. This is just even if I am not completing the remaining slides, I'll just give you an overview. So using the framework that I developed, you can put in that constraint. And then you can actually get to select for only 80 models which satisfy those constraints. Beyond the 80 models that we got, you may need some other experimental information to further restrict down to maybe 1 or few models. Okay. Is this part clear? Okay. So what I wanted to tell you is that the framework where we added the noise allows you, and along with the, sort of the proposition of the MFPT, the mean first passage time as a measure to quantify cell-cell transitions, how easy it is to transition between different states, allows you to do this, okay? So I'll skip this slide. So I just wanted to say one result that one of the reasons we proceeded with MFPT as the measure is because the 5 measures I showed for the quantifying the relative stability, they're very highly correlated with each other. And this we find not only by studying this Arabidopsis root stem cell-ish model, but another model for pancreas cell differentiation. So this result is independent of network structure, presence of other attractors or type of function. So it is not limited if you restrict to NCFs. You can choose any function. These measures seem to be correlated, which allows you to choose one of them to proceed with. So, see, the MFPT also has the following advantage. So we had these 4 cell types, and you can compute the mean first passage time between any of the transitions between these 4 states, okay? And then you have a directed network where what I have not shown is that the ease of transition from QC to CEPL is not going to be the same as CEPL to QC. So they will have different weights on these edges. Once you have this directed graph, actually you can even use our method to further go forward and perhaps theoretically or computationally infer a potential cellular lineage tree if you had all the lineages. And the way to do it, as we propose in our paper, is to take the analog of something in the directed graph, the directed analog of a concept known as minimum spanning tree. So basically you try to find a rooted tree such that the sum of the weights in the tree is minimized, starting from this graph. This graph you can get by computing the MFPT. So for different models, we have this 1 to 75 models, right? They will have different MFPT values. So you will get different trees for them. Okay. And this can be approximated by the cellular lineage tree, and that's what we did. So for the 1 to 75 models, we computed the MFPT between all pairs for the 4 attractors, and then we computed the tree. And you can see there are 13 different trees which we see across the 1 to 75 models. From this tree itself, you can exclude some models which do not comply with the known biology. We know that the quiescent center has to be the least stable. So it has to be on the top of the tree. Okay. So this QC. So of the 13, you can only restrict now to the 4 which are shown. The remaining models can be excluded. Okay. So these are some tools that we have developed for model selection. When you are building a model systematically for developmental gene regulated networks, restricting to the Boolean framework. Is this part clear? I don't want to go into the details. So one of the other developments we did over the 2016 paper is that how do we compute the MFPT? Actually, it is computed by first computing this fundamental matrix Z. And this Z is a matrix of size 2 to the power n cross 2 to the power n, where n is the number of genes. Okay. So as soon as you go for n equal to 20 or 25, then doing the exact computation in a MATLAB becomes, prohibitive. So we actually in the paper also give a stochastic formulation to approximate the MFPT for larger networks, which allowed us to scale up and study networks of sizes 20 and higher. And we have shown that the exact MFPT by doing the matrix computation is very close to the value that you get with the stochastic version of the algorithm. So I'll just quickly end in 5 minutes by showing this, the next part of the story. So the one thing which basically intrigued me is that when these Boolean models were constructed by various authors for developmental gene regulated networks, in the papers they had never mentioned if their stem cell state or the progenitor state is less stable than the differentiated ones. So the question which arose is that have the authors who have constructed these models accounted for this property? That is, as I said, maybe as George Burke said, all models are wrong, but some are useful. So when you build the model, you are trying to capture some properties. Was this property of relative stability accounted for? And to sort of investigate that, what we did is that Alvarez-Bulia's group since 2010 had built 3 other models over the years, which had increasing complexity and more nodes. So what you see is that their 13 model, like their 9 model, actually complied with the known relative stability, where quiescent center is actually the least stable. You look at the sizes of the basin of attraction or the minimum spanning arborescence, the lineage that we inferred. But their latest model in 2020, if you see, no longer had that property. That is the the least stable state is no longer the least stable that you expect from biology. So it also begs the question what was happening over the years while they were improving the model. So these complied, these did not. So I believe that whenever the constructions have been made for Boolean models of developmental gene regulated networks, the authors have never systematically taken this constraint into account, which is inherent or basic to any developmental process. So then we went ahead with the following challenge. Can we find Boolean models that will recover the correct relative ordering? That is, can we improve this model? So we'll we wanted to try out that we will not fix the structure of the network, that is, we'll keep the connections same, but can we just change the functions, the possible functions, which will fit the data and get the correct ordering of the attractors. And actually we developed an iterative algorithm for that, greedy algorithm. I'll just give you the feel of it. So like I did for the 9-node network, you can ask how many different combinations of other functions which can give you the 6 attractors which are there by the node network. By restricting to even the NCFs and ROFs, the number of possibilities which or combinations which give you back exactly the 6 attractors, maybe some additional ones, is already 5.43 into 10 to the power 10. Unlike 1275, which I was able to do there exhaustively, now I cannot do exhaustively. Okay, we cannot characterize each of them exhaustively. So now, in the search for a better model, which may comply with the relative stability, the known relative stability, we designed a iterative greedy algorithm in the paper, where basically we start with the model published in the paper. We change functions starting in some smart way at different nodes and see if a single change in the function improves the relative stability over the existing model. Luckily, we did 1,000 such runs starting randomly at some place, and in 990 of those runs we were able to find at least one answer which is better and actually is basically complying with what you expect in reality. Okay, so with that I'll summarize. I mean, the paper has many results. You're welcome to read it if you're interested, but the crux is that we have given a systematic framework to basically capture the metaphor of Waddington landscape within the Boolean modeling framework, and you can use the tools, further the tools that have been developed along with MFPT, the stochastic version, trying to infer a cellular lineage tree for model selection. Yeah, so I hope that people use these tools when they are building models for newer systems which have not been explored further. So with that I'll end. So I'll thank all the people, various collaborators, and in particular the students who did the work, and also the funding agencies which have really supported us over the last 10 years in the institute. And I think I'm on time. If there is any time, I can take questions, or otherwise I'm around today and you're happy to ask me questions.
So I have a question regarding the application of these Boolean networks when it comes to development. So a lot of times when you look at development, it deals a lot with gradients more than, say, a binary, so presence or absence, but it's more of a gradient of, a specific transcription factor, for example. So how, if you're applying Boolean networks to such processes, how exactly does a network capture that? So is it like maybe something to do with the mean field passage time, or?
We have not gone there, but for example, one of the successes of Boolean modeling has been a model by Rekha Albert's group to explain a developmental process in Drosophila. So one of the ways they do it is that you can actually think of it as a spatial thing, where you basically have a lattice, or things which are connected. So you can think of cell-cell communication as well. So you have a Boolean model in one cell which communicates with Boolean model with a neighboring cell. So you can have a kind of a grid, if you say, and you embed models in each of the grid, and that has been tried as well. So okay, but I have never dabbled with them. So one has to still explore further. So I don't see many examples of that. Although that paper came out in 2003, many such attempts further down the line, to be honest. And I have myself not dabbled with it. That model also has the other complication that some nodes have more than one discrete states. Okay, so yeah. Thank you.
Marcin.
Thanks, Arijit, for the talk. Just a quick question, like when you show these different functions that you find in biological Boolean networks reconstructed, most of them had this odd bias. Is it just because nested canalizing functions have odd bias, or there is more to it?
No, so I think yeah. So one thing is clear that both read-once functions and nested canalizing functions are odd bias, and since we were systematically looking at it now in the hindsight, it's not a surprise. But yeah, that seems to be the answer. But the other thing which I wanted to say is that, I mean, it's very funny. I mean, so there are still some even bias functions, okay? Turns out that if you look at them carefully, they're modeling errors.
Sorry?
They're errors by modelers. So this could be also a signature that, when we see a even function, when we look at it, actually, people had to build the model, whatever was available, whatever fitted. They didn't explore the all possibilities like we systematically explored. So when you look at them, some of them are ineffective functions which have been assigned there. So this could also be used as a sanity check for the modeling purpose. Yeah, so I think it's an NCF, but it has some interesting thing. If you have an even function, could it be a modeling error?
Like some way to, I don't know, check for artifacts.
Exactly. So we clearly see those many of the examples when we looked at it, they were actually seems to be errors.
Right.
Thank you.
So any further questions?
So in the second part of your talk, where you discussed this hopping to different attractors, you were allowing that by the addition of noise.
Yeah.
So I was just wondering, I didn't quite get, were you also evolving, were you allowing the system to evolve the attractors as well? Or were the attractors already fixed?
So the attractors were fixed because, so basically you have the model and the attractors known, right? Because the attractors are already the steady states of the model. And again, I wanted to emphasize, many people ask why not synchronous, asynchronous. Actually, the type of update may not matter. Even in the asynchronous case, I had a comment yesterday on the slide that in the asynchronous case, once you reach the attractor, you cannot go out of the attractor. So again, the asynchronous case is not ergodic as well, first of all. So as far as the attractors we are going to model in development, typically they are going to be fixed points. So it doesn't matter which update you take. Now, what we wanted to do is that, given the attractors and the model, how do we quantify the relative stability and the cell transitions?
One more follow-up question. So you mentioned that you have a trick of designing these attractors?
Yes.
I'm just curious, is it similar to the way Hopfield designs the attractors?
No, it's very simple actually. I mean, it's not a big deal. I mean, I can tell you here. So I'll just give you the crux of it, and it's very simple. So let's look at the node JKD, okay? Okay. So JKD is controlled by two nodes, SCR and SHR, right? So this is going to be a two input function, okay? And look at your attractors. So let's look at the combination. So if you look at the first attractor here, so these attractors are given. So when SCR is 1 and SHR is 1, then JKD is 1, okay? So basically you are writing a two input function. So you basically have to so these are the four rows, right? So if you see, based on the first attractor, this is already fixed. You can't do anything with it. So basically what we are trying to see is that how many possible functions out of the 16 possibilities can fit the four attractors. So similarly, if you look at the second one here, when SCR is 0, SHR is 1, JKT is 0. This gets fixed. Okay, you can't play with it. And then if you see SCR is 1, SHR is 1, JKT is 1, that we have already got. And then you have 0, 0, 0. So this gets fixed. So these are fixed. Okay. Now there is only one row in this truth table which you can play with, and there are two possibilities. So there are two possible functions which you can fit data with. Actually, when you do this, you may also find in many cases that the attractors cannot fit the architecture. Then you have to change the architecture. Okay. So this method actually was again developed with Olivier Martin in a paper in 2011 by us actually, and this is a very simple trick where you can actually get. So what we are able to estimate is which are the combinations of functions which will give you the 4 attractors. They could have additional attractors as well. Okay. So this trick is very quick to figure that out. Again, it can be used as a modeling trick. Sitabro.
Okay, sure.
I don't know if this is done in other contexts. But
No, I mean, because I guess this is solving, right?
yeah, sorry.
Yeah, so because you mentioned Hoffield, so I remember this discussion I had with Olivier. So we had a similar work at that time on Hoffield model where we changed the architecture of the Hoffield network to make it modular, and we found similar behavior in Hoffield model. And Olivier said that I really envy you because in Hoffield you can actually, here you actually have to find the attractor by doing simulations.
Yes.
In Hoffield, because you are constructing the weights, knowing what attractor you want, you actually don't have to go out and find it. You already know which are the attractors, and so you can do this analysis by knowing exactly what the attractor should be and when you don't get there and so on. So that's a big advantage in Hoffield because you can actually construct the network according to where you want the attractors.